One.
Hi, welcome to Friends of Brain Injury.
What about entire cells, like bowel cells or bone marrow cells, liver cells? How do they go about copying themselves? This process of asexual reproduction, or mitosis, DNA is responsible to manage cell division or reproduction. Routinely, a cell's DNA is found partially unraveled so that portions can be accessed to produce the proteins. Histones, protein structures, act like spindles for winding and unwinding the chromosomes that are responsible. When a cell needs to divide itself, though, it undergoes a specific process to organize the chromosomes' alignment to make cell division more efficient. To understand the process of cell division, it's best to break it down into stages, just like we did for protein production. Some aspects of cell division aren't too different from protein production, as we make a copy of a cell and it involves copying the proteins, just at a much larger scale this time. Remember, mitosis is day-to-day cell division. Different cells undergo mitosis at different rates. Some cells undergo mitosis frequently throughout their life cycle, and other, usually highly specialized cells, only do it for a short period during their early growth. And they never really undergo mitosis again. Medical research is ongoing how to encourage these cells to resume mitosis after completion. So what happens in the average cell and say the small bowel needs to divide? That's what we're talking about here, not about the production of sperm, eggs, any of that kind of thing. First, though, before we pass through the stages of division, a cell needs to make preparations to divide. So we call this initial stage interphase, and interphase is subdivided into stages too. So remember, this cell is busy making proteins. Day to day, doing cell kinds of stuff, being awesome, secretions, absorbing nutrients, making enzymes, just being an awesome small bowel cell. The cell needs to divide. It will need to divide from the initial cell, the so-called parent cell, and that's the cell that will do the dividing. And it will yield then, when it divides, to what are called daughter cells, even though they kind of share a lot of initial organelles and things. The first stage division, which we said was interphase, divide into three subphases. G1 or gap 1. This is where the cell will commit to cell division or not. It's here where the cell will have to produce enough enzymes, other organelles, and have enough materials in order to make that decision to divide or not. It is here where it will produce the necessary proteins to make DNA duplicates, if it has enough. During this phase, it's essential to preserve the DNA, no mistakes. There can't be errors here, or else again it'll stop. So let's go over the entire process. DNA replication starts once the cell completes G1, that is, to produce the necessary proteins, organelles, everything that it needs. If it can't, it will not progress forward, and the cell may even self-destruct, depending on. So, second stage, which is, remember, we're still in interphase. G1, S, for synthesis. Replication of DNA will start at multiple sites all over throughout all of its chromosomes, because it's got to copy all of them. So multiple sites of chromosomes will start. An enzyme called helicase will unwind the DNA at various sites. Now we're dealing with something else. And it starts what's called a replication fork, where DNA is to be copied exactly as it is. We're not just making a few proteins here. We are copying everything. So many replication forks are formed as this process starts in multiple places on multiple chromosomes. A special type of RNA called RNA primer is synthesized by an enzyme called synthase. An RNA primer will serve as a starting point to actually copy it all. Then you get elongation. DNA polymerase adds new nucleotides in the 5' to 3 direction in the original DNA as a template. You get what's called a leading strand as a DNA strand, which is synthesized continuously as the replication fork moves forward as the DNA polymerase replicates. The lagging strand behind is synthesized discontinuously, so on, off here again, into short segments called Okazaki fragments. And they start with a new RNA primer as they pick up and continue to replicate in pieces. Okay? Termination. The RNA primers will cut loose at the end of the DNA segments they are coding, and DNA nucleotides will be inserted. So it's not a smooth process as far as the lagging behind strands are concerned, the lagging strands. An enzyme DNA ligase joins the Okazaki fragments together into a single then continuous strand. So it doesn't start out continuously, but that happens as it fills in. At the ends of DNA strands, the 5' end cannot be replaced. So the enzyme telomerase, keep telomerase in mind because it does affect aging, we think, extends the strand, allowing the other end to be filled in. Then the end result is 2 identical DNA strands which are now ready to go through the process of cell division. Remember, this is about cell division here, so we need identical copies for mitosis. Obviously, there's more to it than we just explained. We skipped some very detailed steps and some enzymes as well. If you're interested, we can go into much more depth, but this gives you what you need to know. All right, so finally, once the DNA is copied, you've got identical copies of all your chromosomes. They had to be unwound, and then they have to be rewound, but we'll get to that. Finally, G2, the third and final step of DNA replication. So the DNA pairs form back into chromosomes. They wind back up, they pair up and line up, and the cell will enlarge and it will take on what it needs to make itself big enough to get ready to divide. It will enter what's called prophase next. And the pairs of chromosomes, now we've got dual pairs. Now we've got, not dual pairs, but pairs. These are called sister chromatids, is the term for that, because they are identical, they're twins. They have to start to line up so that when the cell divides, there's one copy in each, okay? This subphase isn't completely understood, but we do understand quite a bit about it. Some go right from synthesis right into the next phase. Others kind of hold here for a little bit, do a checkout, make sure everything is good, and they move into prophase next. During prophase, the histone condenses the DNA tightly, and the sister chromatids, which are identical copies, as I said, they are joined by a structure called a chromatid. And this, at the center, this chromatid, these paired chromosomes have a centromere. I apologize. The sister chromatids are joined by a centromere. So the centromere then, which binds the two together. Then another structure, the centrosome, will start to migrate to opposing ends of the cell that's going to divide. These centrosomes will help to pull them apart. The centrosome produce spindles by the polymerization, that is chaining together. Remember what I said about polymers being long chains? Of what's called tubulin. It is a protein that it can be pulled, shortened by motor-type molecules. All you have to know is that these centrosomes, these centrioles, their job is to pull apart these now lined up chromosomes, these pairs, these sister chromatids. Their job is to pull them apart, and they do it like acting like a spindle, both sides, here. We don't know like the very small details of this, but we generally understand. So of how it looks, generally how it works. So in prophase, the DNA is now wound up. It forms sister chromatids. The centrosome containing the two centrioles will send the centrioles to opposing ends, and converts this protein called tubulin into form kind of like a spindle to help pull apart. And then we get into prometaphase, the nuclear envelope, which has been protecting the cell nucleus containing these paired, it's still there in the middle with the lined up structures. That breaks down now, and the microtubules grab on to these paired chromosomes, one on each side. So remember, they're lined up, paired, and the tubulin grabs onto the centromere at a specific protein, a protein that's kind of for this purpose, to pull them apart. Okay, so these spindles are driven by a molecular motor that's powered by ATP. We talked about ATP being the energy source for cells. So it's going to run this little motor that will pull the chromosomes apart. The polymerization and depolymerization of the kinetochore, which is that protein handle that the spindle grabs onto, will work together to move the chromosomes as we progress. So that's what prometaphase is. Nucleus is gone, and the spindles now grab on to each chromosome's handle, kinetochore, and they will start to pull them. Metaphase, the chromosomes are pulling the pairs of chromosomes first into alignment, and these centrosomes are found on opposite ends of the cells and will move them along the equator of the cell, the largest portion of the cell in the middle. So they will align them now first, and then they will start to pull. This is another cellular division checkpoint. If the chromosomes are lined up along the equator, the other name for this equator is called the metaphase plate, just a name for this structure, and the kinetochores are connected, the cell may proceed. But if something is wrong, if there aren't both pulling on the kinetochore, that handle, maybe it's got two of them, that's bad. You don't want excess chromosomes in one cell. This checkpoint may trigger the destruction of the cell, or maybe it won't. This is a hint for future genetic problems. Or maybe it's going to pull a piece and a half. This is also the source of potential genetic problems. Mainly in cells like this, that leads to cancer or other problems. But when it comes to sex cells, that leads to other disorders. All right, so if it passes the checkpoint, it finally moves into anaphase. Anaphase is broken into two phases, anaphase A and anaphase B. The cohesins, which bind the chromatids together, which is like a protein structure that holds it now, breaks. And there are now two separate but identical chromosomes. Instead of being bound like this, they're now broken, and they're ready to be pulled apart. The microtubules will start pulling them towards the opposite sides of the cell. They're no longer bound. They're not sister pairs anymore. They're now going to start heading the opposite direction. Anaphase B, other microtubules will push against one another, causing the cell itself. So we've got pulling and pushing. The cell is now starting to, look like this. We will discuss these types of errors that can happen, non-disjunction repetitions, in another unit. But these proteins, there are also proteins that act like stop signals, and the centromeres that assist in ensuring proper connection guidance. This is an important function also as a cell continues through division. At the end, then, is telophase, where it reverses what happened in pro and prometaphase. The tubules will shorten and shorten and shorten because of the ATP-run motor. They disconnect from the chromosomes. The microtubules lengthen till the cell is the correct size. A new nuclear envelope will form. Finally, the chromosomes decondense back into what they were during interphase. Division is complete, and the cell may not be completely done internally, but at this point, you got two cells. Now, with cytokinesis, which is not a phase of separation, the cells kind of break away from other cells. A cleavage furrow has formed, and there's really two bound cells. They have the contractile ring. The contractile ring is where the metaphase plate used to be, the equator, and the chromosomes are not, they've separated from each other for the cell, and now you've got two daughter cells. And where the ring is pinched off, the cell membrane is closed. The two cells are separated into two daughter cells. Plant and animal cells don't always undergo cytokinesis, just something to keep in mind. After the two daughter cells are completely divided and go into their own kind of thing, the chromosomes unwind. They produce proteins as the parent cell from which they're derived did before. In some situations, the cell may have to prepare to divide yet again, especially if they're immune cells or cells in the digestive tract or cells that repair damage. Other cells may only divide a few times and never divide again. Specialist cells divide very few times, as their specialty costs them that ability to divide as they become more specialized. Let's take a break at this point, and the next episode we will discuss a specialized type of cell division. It's called meiosis. This type of cell division, is found only amongst sex cells and allows increased diversification traits in individuals. So, please remember, click that bell icon if you found this helpful. Click like, subscribe. We'll see you soon. And share this with anyone who may benefit. Also, check out our Friends with Brain Injury, on Facebook, and look for our website of the same name. Hopefully you'll find helpful information there, and we will be moving on to the next chapter shortly. See you then.